Good morning students, I am Shomajit sir. Today I am here to deliver my lecture on topic proteins. This is from the chapter biomolecules. So watch the entire lecture to get basics out of it. So without any delay, let's start with the topic proteins. So let's start with the topic proteins. This is falling under biomacromolecules. So what are biomacromolecules? These are biomolecules having molecular weight greater than 1000 Dalton. Alright. So what are biomacromolecules? These are biomolecules having molecular weight greater than 1000 Dalton, and they include proteins, polysaccharide and nucleic acid. Alright, so in this video lecture we'll discuss about proteins, and their molecular weight is 10,000 Dalton and above. Alright, so their molecular weight is 10,000 Dalton and above, and they are acid insoluble fractions, that is macromolecular fractions, and it includes what? Macromolecules from cytoplasm and organelles. So it includes macromolecules from cytoplasm and organelles of a cell. All right, so you can see three diagrams are there, one of protein, one of polysaccharide, another of nucleic acids. So you can see in a biomacromolecule, you can see the structure of cell membrane. All right, so you can see lipid is not strictly a macromolecule, as its molecular weight does not exceed 8000 Dalton, but it comes under acidic. All right, so it comes under acid insoluble fractions because many lipids are arranged into structures like cell membrane. All right, so it comes into acid insoluble fractions because many lipids are arranged into structures like cell membranes. And when a tissue is grinded, all right, when the tissue is grinded, the cell membranes are broken and form water insoluble vesicles, and they cannot be filtered along acid soluble fractions. Understood? So this lipid is not strictly a macromolecule, as its molecular weight does not exceed 800 Dalton. Alright, again I repeat, 800 Dalton. So you can see the structure of cell membrane and the elaborated structure. You can see the internal structure of cell membranes are there, where lipid layers are there, transport proteins are there, phospholipids are there, carbohydrate chains are there, proteins are there. Alright. Now, in a biomacromolecules, the average component in a cell, the average composition of cell, we can find water, we can find 70 to 90%, proteins we can find 10 to 15%, carbohydrates we can find 3%, nucleic acid we can find 5 to 7%, and ions we can find 1%. Alright, so these are the compositions of carbohydrate, protein, water, nucleic acid, ion present in a cell. Alright. So today's topic is proteins, that is 10 to 15% present in a cell. So in biomacromolecules, we'll start with proteins. So you should know the unit of protein is amino acid. All right, so the unit of protein is amino acid, and proteins are heteropolymer of amino acid. All right, so as you all know that amino acid is a unit of protein, and proteins are heteropolymer of amino acid, and they are polypeptides. All right, that means that all the amino acid are joined together by polypeptide, by peptide bond. All right, and linear chains of amino acid are linked by peptide bonds. So all the amino acid chains, all the amino acids are joined by peptide bond to form the protein. All right, and peptide bond is formed when a carboxylic group of one amino acid reacts with the amino group of the next amino acid by releasing a molecule of water due to dehydration. So you can see on top chains of amino acid are there, A1, A2, A3, they're given for example. All right, so for a amino acid, you can see for amino acid, one side there is a amino group, another side is a carboxylic group. All right, so when this, you can see when from the carboxylic group OH and from amino group H is removed, that means is lost, that is dehydration takes place due to loss of water. The peptide bonds are formed and all the amino acids are joined together to form a protein. You can see below peptide bond is shown, and on the left hand side below you can see the amino acid and peptide bonds are shown. So all, again I repeat, all the amino acids are joined by a peptide bond and these combine to form a protein. So in amino acid, the carboxylic group present forms an acidic amino acid, and if amino acid is more, if amino group is more, then it is basic amino acids, and if carboxylic acid is more in amino acid, it's called acidic amino acid. So now you can see the functions of protein. What are the different functions of protein? You can see it is used for growth and tissue repair. Again I repeat, it is used for growth and tissue repair. It is also used for transport nutrients across cell membrane. All right, it is used for transport membrane across cell membrane. Example, GLUT4 enables glucose transport in a cell. It also acts as intercellular ground substances. All right, it acts as, again I repeat, intercellular ground substance, not intracellular. It is intercellular ground substance. Example, collagen. It also acts as antibodies to fight infectious organism. All right, so it produces antibodies to fight infectious organisms. It also acts as receptors. Example, receptors of smell, taste, hormones. And there are some hormones, example insulins are there, some enzymes are there, example trypsins, and some pigments are there, example hemoglobins. All right, so there are, and those, there are very important functions of protein. Protein has a very important functions in our body for growth and tissue repair. It acts as a receptor, it acts, some proteins are hormones, some proteins are enzymes, some proteins are pigments. All right, so you can see insulin is there, trypsin are there, hemoglobin is there. Structure one in left hand side you can see in the figure. Insulin structure is given, then trypsin structure is given, then hemoglobin structure is given. Next you can see these are two important words, the most abundant protein in animal world. If question comes, name the most abundant protein in animal world, you will say collagen. Collagen is the most abundant protein found in animal world. And if question asks, name the most abundant protein present in biosphere, that is rubisco. Rubisco's full form is ribulose bisphosphate carboxylase and oxygenase. So again I repeat, the most abundant protein in animal world is collagen, and the most abundant protein in biosphere is rubisco. It's ribulose bisphosphate carboxylase and oxygenase. All right, it's ribulose bisphosphate carboxylase and oxygenase. Now you can see the structures of protein, or different levels of protein, structural levels of protein. There are four different structures of protein. One is primary structure, secondary, and tertiary and quaternary. All right, they are moving from simple to complex. So primary is the most simple structure and quaternary is the most complex structure. Okay, you can see a Haim structure is given on the right hand side. Now moving to the primary structure of protein. Primary, alright, it will be a straight structure, a one single chain will be there. Alright, a chain of amino acid will be there, it will be joined by peptide bonds. And you know that in amino acid, one terminal will be an amino group and another terminal will be carboxyl group. So it describes the sequence of amino acid, that is positional information in a protein. So left hand, alright, left hand side, or the left end is the first amino acid, that is N terminal amino acid, and the right side you can see the last amino acid, that is C terminal amino acid. So in the structure in the right hand side you can see left side is the first amino acid, that is N terminal amino acid, and right side is the last amino acid, that is C terminal of amino acid. So primary structure of protein is a straight structure. You can see on top one straight chain is there, and this chain is made up of numerous amino acids joined by a peptide bond to form a protein. As I've said, the unit of protein is a amino acid. All right, so you can see in the structure amino acid and then peptide bond, the figure is given. Now moving to the secondary structure of protein, alright, or secondary level of structure of protein, you can see here one or more polypeptide chains are folded in the form of helix, alright. So here it's not a straight structure, here it is not a straight structure of protein, here it will be folded into a helical structure, or it's called alpha helix, or it will be folded into beta pleated sheet. You can see in the right hand side it can be folded in the form of alpha helix or beta plated sheet. So here one or more polypeptide chain are folded in the form of a helix. It has only right handed helix. Example, keratin, fibroin, that is silk fiber. So example of secondary level of protein is keratin, fibroin or silk fiber. So these are not straight chain structure, these are folded into a helix or beta plated sheet. So that's why examples are alpha helix and beta plated sheet. Two different types of conformations or folded as seen. Alright, so it is right handed helix for alpha helix and beta plated sheet. So you can see a typical strand of hair. You can see keratin is there, strong links are there formed by disulfide bond, and weak links are there formed by saline and hydrogen. Alright, so fibroin or silk protein, you can see anti-parallel beta sheet rich in alanine and glycine. ALA means alanine, short form of alanine is ALA, and short form of glycine is GLY, and small side chains allow close packing. Alright, so you can see the keratin structure on the left hand side and the silk fibron or silk protein. You can see these are the secondary level of protein. Now moving to the tertiary level of protein, here you can see the helical polypeptide chain is further folded like a hollow woolen ball, and it gives 3D view. Alright, and tertiary structure is necessary for many biological activities of protein. Example, myoglobins are there and enzymes are there. Alright, so example of tertiary level of protein is myoglobins and enzymes. It gives 3D structure of protein and it is folded into a woolen ball. Alright, so you can see the secondary structure, alpha helix and beta strands are there. So you can see the myoglobin structures are given. Alright, so as you all know that all enzymes are made up of protein, isn't it? So this is the tertiary level structure of protein. Now moving to the last structure of protein, that is quaternary structure. So here in the quaternary structure, you can see here more than one polypeptide chains form tertiary structure, and each chain functions as subunit of protein. Alright, here more than one polypeptide chains are there. Alright, example, hemoglobin, it has 4 subunit, 2 alpha subunit and 2 beta subunit. Alright, so again I repeat, in the quaternary structure there are more than one polypeptide chains. Alright, and each chain has subunit of protein. Your example is hemoglobin. We know that inside our blood there is RBC, and inside the RBC the hemoglobin is there, which carries oxygen to different parts of a cell. So again I repeat, in the primary structure it was straight, in the secondary structure it was folded into a alpha helix and beta plated sheet. In the tertiary structure it is folded to form a 3D structure, woolen ball like structure. Right, like you can see fibroin. Right, fibroin is there for the secondary structure, and in the 3D structure, tertiary structure, you can see myoglobins, and the quaternary structure you can see hemoglobins. Alright, so again I repeat, the primary structure, you can see primary structure is a straight structure having one strand, having numerous amino acids joined by a peptide bond. Alright, this is the primary structure of protein, where straight amino acid chains are there with peptide bond and forms a protein. Alright, So in the secondary structure, you can see they are folded into a alpha helix and beta plated sheet, example keratin and silk fibrin. And in the, so these are the example of secondary structure. And the tertiary structure, you can see myoglobins are there. One strand only folded into form a tree shaped, and more folded are seen to form a woolen ball shaped. And in the quaternary structure, there are more than one polypeptide chains, and each chain function as a different subunit. Alright, so here example of quaternary structure is hemoglobin. Now you can see proteins at a glance, like all the structures are given: primary structure, secondary structure, tertiary structure, and quaternary structures are there. This slide is very good, very interesting. You can see one straight chain, that is a primary structure, chains folded into helix, a small alpha helix, and a beta plated sheet in the sheet format, is beta plated sheet in the structure of. And tertiary structure is polypeptide chain giving a 3D structure, and quaternary structure more than one polypeptide chains are there, so that is called assembled subunit. Right, so you can see the structure: primary structure, secondary structures, tertiary structure, and quaternary structures, and beta plate. Everything is given here at a glance. It's a very interesting slide. These diagrams are very important. You will copy this down, and all the examples of all the structure of proteins are very, very important. You have to memorize this examples, how they are formed, and what are different types. Thank you, student. This was all about the entire video lecture. We have discussed about the topic proteins. This from the chapter biomolecules. Hope you have understood the video lecture. So stay at home, stay safe. I'm Shomji sir, signing off for today, and again we'll meet in the next class. Thank you, students.